And also from my side, a warm welcome to EFSA and to Parma. And a warm welcome to the first session. We're starting now our first session, and the name of the session is NAM's Global Landscape. And this is following a tradition from the GSRS conferences, always to start off with a global view of what is happening in the different member countries of the coalition, the main achievements and the new discoveries. And our journey here will take us straight away to the US, and particularly the FDA, and will be by Dr. Christy Muldoon-Jacobs, who will be his first speaker. Christy is the acting director of the Office of Food Additive Safety, also known as OFAS, where she directs the office responsible for overseeing the safety of ingredients in foods and substances that come into contact with food, either as sort of packaging or manufacturing. With no further ado, I would like to invite Dr. Christy Muldoon to come and give a presentation, the title of her presentation being Technology for Future Smart Food. Thank you.
Good morning, everyone. Thank you for the kind invitation, and thank you for having me here today. I think it's been noted we've been here for a couple of days already, and it's been some really exciting conversations, starting with Ilmarak and hearing what's happening and what are priorities and some commonalities that we all share as we want to move and advance in this area. And being the first speaker of the first session and hearing a lot of inspirational talks about where we all see ourselves going and the vast responsibilities of FDA generally, I think I'm going to bring us back a little bit down to earth in terms of from the programmatic perspective as it relates to the foods area, what we are doing, what we are seeing, and how we're starting to deal with and incorporate some of the innovative products, some of the innovative technologies as it relates to our programs and our responsibilities related to safe foods for the U.S. food supply. Can we bring up the slides? Maybe in a minute, maybe less. So as we are waiting, I think it was mentioned, so the Office of Food Additive Safety in CFSAN, soon to be part of the larger Human Foods program, has a really broad responsibility. And I will touch on this in my talk, but while we're waiting, I think it's really a powerful and a unique approach where we're bringing together experts who understand the food additive regulations and the definition of how food additive regulation in the U.S. really ties things together that are considered direct, indirect, and new product foods. And so this is within our program, our purview and our responsibility in the Office of Food Additive Safety. And I'm trying to fill time as best I can while I'm waiting. Anybody know a good joke? All right, I think I'm going to start. Luckily, I brought my handy computer up here with me. And so we'll have the slides catch up and I'll get started because I know we have a very busy day and I don't want to hold us and put us behind schedule before we even start. I'm sure organizers would appreciate that. So as I was touching on, the regulatory space of CFSAN is large, and I had mentioned food additives, but in addition to food additives, when we're talking about the safety of foods, we have responsibilities for other things beyond food additives. We're responsible to ensure food as consumers. Here we go, slide number 1. We can go to the next one. There are the responsibility of SIPSAN is broad as it relates to food safety. We're looking at not only the safety of food additives and food context materials, as I mentioned, but we're looking to ensure the safety of, to control things like contaminants, right? To understand and make sure that macro and micronutrients are there at levels to provide for nutritious foods. We're also looking at things like dietary supplements, and there's innovation in products and space for all of these. We've got different categories of definitions. We've got food additives, color additives, but also GRAS substances, something that I think people hear quite a bit about and is often largely misunderstood, I think, within our regulatory system. Can we go to the next slide? Oh, I have the controls. Aha! I can, here it is. Hold on a second. Oh, here is the big button. Okay. So, given that this is the global regulatory, right, meeting, I wanted to not spend the entire time talking about regulations, but just in the context of this presentation, two, I think, important parts of the U.S. Food, Drug, and Cosmetics Act that give us our jurisdictional authority and connect important parts of our law that connect our mission to the safety of the food and the programs that we execute to do that role. And this one part that we refer to as Section 409 of the FD&C Act defines a food additive. And it's really important to note that this definition of a food additive is quite broad, because what's included in a food additive, and we don't need to read through this whole thing, is that it includes substances that are intended to have a technical effect in food, but also expands beyond that to anything that might be intentionally added with an unintended presence in food. So that expands to anything that would be used in the manufacturing of food or the manufacturing of packaging used for food. And these are all included within the definition of a food additive. It sets out responsibilities for authorization of food additives that have to be authorized through regulation. Importantly, it also sets the food safety standard for anything that is intentionally used or may become a component of food. It does set out some exclusions from that definition of what requires pre-market authorization. But tied to this definition is the requirement for anything that is becoming a component of food through its intentional use to meet the same safety standard. And because of this tie within 409, we conduct safety standards for anything that is intentionally used or could become a component of food under the same safety standard, which is a reasonable certainty of no harm under the intended use conditions. This definition in 409 and its exclusions are important to tie to Section 402, and I promise this is my last regulatory slide. But this is important because it establishes regulatory jurisdictional authority for what the FDA can do and actions that we can take for adulterated food. When there is a food that is found to be adulterated, the FDA has broad authority to take action to remove adulterated food from the market. And why does this connect to 409? Because within the definition of an adulterated food, it says any unapproved food or color additive, food that contains any unapproved food or color additive is deemed unsafe. So because GRAS connects to the definition and sets a regulatory standard, and because anything that is not an approved food additive and therefore does not meet the safety standard can be adulterated. And so this gives us a lot of ability to look at anything that would become a constituent of food through its use to determine its safety. So how do we do this work? Because this requires a presumption that before something's used in food, you have to be able to meet that safety standard, whether you be the regulator that's authorizing it, you being industry that's using it. So what do we do at FDA? Within FDA, we have the Office of Food Additive Safety, where I am the current acting director within that office. And we have several programs to administer these authorities at a pre-market stage based on these different categories. Whether something might be a direct ingredient, macro or micro ingredients that are specifically added as food additives. We have a food additive or a color additive petition. Small little asterisk star is a color additive, cannot be considered GRAS. So any new thing that imparts color to food, whether it be a natural or a synthetic color, has to come through an authorization through a color additive petition. These are generally done through a food or color additive petition, but we also administer a GRAS notification program. And this is because to be able to meet the safety standard for GRAS, there needs to be publicly available data that scientists with experience would agree and determine that that use is safe under the intended use conditions. At FDA, we think we have expertise. And so as industry is evaluating their substances to determine whether or not they meet the safety standard for GRAS, we have a program where they can come in and we can also look at the information that they have provided to make that safety decision and see if we would have any questions about their conclusion as well, whether we would come to a situation where we would say, we think that there's questions here that need to be resolved before we would issue a final, what we call a no questions letter on that safety determination. So those are evaluated through the GRAS notification program. Then we have another program where we really authorize indirect food additives. So these are any food contact substances. So these are substances that are used in packaging of food, as well as substances that are used in the manufacturing of food. These are indirect additives. And then we also have a few consultation programs where we're looking more at whole foods. Now, we don't generally, we don't regulate finished products. That is not the system in the US. We look at the safety of the ingredients to be used in finished products. But as we're looking at plants made from genetically engineered, using genetic engineering or genome editing, what we're assessing is whether or not this raises any safety questions or whether the use of this technology may introduce other substances that need to come through one of our other regulatory programs to show that these are safe. And then many of you, I'm sure, have heard that we've done 2 recent cell culture consultations where we've completed looking at the use of new cell culture technology to generate food made through the use of cell culture. What is interesting and why I'm talking about all of our programs, which I am incredibly proud of, and I find them to be incredibly effective and efficient in ensuring the safety of foods in the US, is that we are seeing that technology and innovation have been a part of these programs since their inception, right? Everything was new at one time, so innovation has been part of the legacy of the program. So how do we do this work, which I'm telling you we've got a long experience and expertise in? We do this through scientific expertise that we have in-house at FDA. Whenever we review any submission, we bring together at least the core teams, which always include interdisciplinary scientists, including a chemist, an exposure expert, a toxicologist, and a regulatory expert. And these teams work together to look at the data and information that's available in any of our submissions. Because innovation, science, questions don't always just hit on these 3 core disciplines. We also have expertise in other areas. We have microbiologists, nutritionists, statisticians, pathologists, clinical data reviewers when we need them, to call on and bring in that expertise to ensure that we've covered the relevant discipline. And this is becoming more and more important as we're seeing evolution in this space. These experts with experience and education and training come together to evaluate the full submission. And the approach that they take, though, for any of these categories is generally the same. We're looking, when we look at these submissions, at when we're evaluating a substance, critically important is what is the substance? What is its full identity, the properties, the composition? What is the manufacturing process? Are there anything in that manufacturing process that could be critical for safety, and how are we looking at that? The specifications, limitations on impurities and contaminants to ensure that those are present at reasonable levels where they may be, or acceptable levels where they may be. The next part of the assessment comes to intended use. Intended use is fundamentally important. I think as anyone who has expertise in this field, we know that the dose makes the poison, right? So just because something is safe under one condition, it might not be safe under absolutely every condition. So we're looking at what do they expect to use it for, and how that would influence the exposure. We build in what we call worst case assumptions into these. One of the popular ones we like to say is, if you say you're going to use this at 5% in all baked goods, how much could that possibly be if somebody, the 95th percent consumer, was eating baked goods that had this particular substance in it at 5% in every single one of those? To come up with a worst case exposure estimate, we compare that data to the toxicology and safety information that we have to look at what levels would we say we're at a space where we're at a level where there may be an assurance of safety under those intended conditions of use. And this is the approach regardless of the category, whether it be GRAS, whether it be a food additive, a color additive, a food contact substance that we take. We like to say pre-market is prevention at FDA. So how much of this work are we doing? We're doing quite a bit every year. In 2017, we saw a big upswing, but this only takes us back to 2017. We're seeing over 200 a year of these types of submissions that are coming in. When you combine food contact notifications, food additive, color additive petitions, we're seeing more and more infant formula, new infant formula ingredients. As we all know, this is a big topic. The amount of biotech consultations that we're seeing are going up. We've, as I mentioned, seen the 2 new cell culture consultations. Another thing, this gray bar at the top, that one's really dramatic. It's easy to see that there's been quite a shift, is related to recycling. We have a program where we look at material that is going into recycled feedstock to make sure that that recycled material is as safe as the material. This is an important issue, I think, globally, and we're seeing a large increase in these types of submissions as well. So let's bring it back to innovation. So some things that we know about this data is not every single thing here is an innovative product. Some of this represents what we would call re-review. Information that if there is the change in a manufacturing of a particular substance, especially as it relates to the FCN program, if there's a new manufacturer that wants authorization for the same substance, they need to come back in. So a significant, a not insignificant amount of this data represents re-review. Things we've seen before that we're looking at again. When we see it again, we recalculate the exposure. We look at any new toxicology data that's come in since our last review to reach the conclusion again. But we're also seeing a significant amount of innovation in these programs as well. We talk about GRAS, we talk about food additives, we talk about color additives like it's one thing, but we really are, if we were to compartmentalize it, seeing a segment of re-review and a segment of new innovation. When we're looking at the GRAS notification program, we are routinely evaluating new ingredients that are being produced through novel applications of food technologies like genetic engineering, fermentation, bioprocessing. We're looking at these processes, we're looking at the ingredients that come out of these processes. In biotechnology, we're looking at these consultations to evaluate the potential effects of genetic engineering and gene editing. Our unified guidance agenda, you can see we are working on a gene editing guidance right now, and hopefully we're hoping it'd be out this year, but federal government clearances take time, hopefully next. But a new guidance document on plants made from gene editing. We're also seeing things related to nanoscale. We're paying close attention to this issue. We've put out a guidance document again that talks about significant manufacturing changes, using nano as an example of where we would say we are very familiar with this particular substance or ingredient, but through your manufacturing change, maybe there's now a nano component that we need to pay attention to. And so we're looking for these. In the space of food contact and packaging material, really exciting innovation happening in this space. We think packaging is just packaging. But with the drive to recycling and innovation, we're seeing new substances used in packaging so that there can be less plastic use, ease of recycling at safe levels. And food colors, big space. Industry is driving towards more natural colors. Any color, whether it be synthetic or natural, have to come through a color additive petition. So we're seeing more of these natural colors come in and evaluating these through the food additive, the color additive petition program. So lots of innovation that we already have experience with. So hopefully, maybe I've convinced you that this is a program that we think is robust and well-suited to deal with all of this innovation that we heard about earlier this morning with these really broad, motivational, and inspirational opening remarks about where we're going and the things we're going to be dealing with. We find that this strong authority and the broad definition of food additives, what's included, excluded in the programs that we've put in place, are flexible and give us the tools that we need to be able to deal with this as we continue with development, scientific development, to make sure that at the current time we have the tools and the scientific expertise that we need to evaluate this. The last thing on this slide I think is an important one, because with all I've said about focusing on ingredients and what we see in change and innovation is the FDA safety evaluation is really targeted to the ingredient itself and its intended use. The manufacturing method is considered, of course, but it's considered as it pertains to safety. We don't evaluate a method of manufacture on its own. It's that method as it relates to the final safety of the ingredient or the food substance. So we heard a lot about these externalities. So what are we seeing as science is moving, and what's the impact here on our programs and who's impacted? We're seeing biotechnology advancement. We know animal, cell, and tissue culture is really making inroads into the food space. There's a lot of interest here in the food sector and manufacturing, and it's certainly got taking time of our experts and regulators to be able to keep up with this technology and make sure we have the expertise to evaluate it. We're seeing microbiological production of micro and macronutrients. We're not just seeing extractions anymore. These things are being synthesized in bioreactors, synthetic biology, genome editing. This biotechnology advancement's coming to the program, and these are having an impact. We're seeing scientific advancement, information technology, AI's advancement, safety assessment, NAMs, tox testing methods. These things are changing as well. Advances in analytical, especially chemistry. We're seeing detecting of lower and lower concentrations. Those LODs are driving down. What are we doing about that with our exposure assessments? We're looking into that as well. We're seeing new products, right? Not just the applications, but the products themselves are new. We're seeing innovative approaches to essential nutrients, new proteins, new sources of fats. The food industry is looking to make healthier products. Where are they coming up with these ingredients from? How are we evaluating their safety? And then, of course, new ingredient sources and production technologies are coming into the food supply. So where's the impact? There's impact on us as regulators, but there's impact on the food market as well. These changes are impacting innovators, farmers, food producers. Consumers are certainly paying much more attention than they have in the past. They want to feel confident that the regulators have the expertise and the jurisdictional authority to do what's needed to ensure the safety of the food supply. The impacts on trade as well as we move to a more global market, right? It's not one food system. These are all things that are being impacted by this innovation and change. So what are we doing as it relates to research? It's a NAMS meeting, right? So we do have an FDA alternatives a working group, and we are committed to promoting the development and the use, where appropriate, of NAMS technologies. I think Susie Fitzpatrick may be talking about this later, but our definition is new alternative methods, not non-animal methods, not new animal methods, but new approaches, because we're committed to the 3 Rs. We have a research project, we have research laboratories as well as regulators that are working in these areas, and we want to make sure that where these methods are ready, where we are assured of their quality and fit for purpose use, that they are incorporated into our safety assessments. And we're looking to continue to develop those methods that answer our regulatory questions. And so we have quite a lot of models that we've been working on, and these are examples of a few publications that have come out of our labs to investigate, develop, and evaluate the use of some of these alternative methods. We've looked at some in vitro models, cells on a chip. We've had a long-standing program. In silico models, we're seeing use and development of more and more. There's a lot of power in the data that we have. And with the use of AI, we can incorporate and leverage that existing information even more. So we've been working to incorporate that data to generate in silico models, and then to make use of lower model organisms, right, to help inform just mechanism in general and help with prioritization. So what are we doing beyond development of NAMs themselves? Through our alternatives working group and collaboration with our NCTR partners, we are looking at building and incorporating existing knowledge to build new tools. And this really will be a thing that will help, in addition to the NAMs, modernize the food supply. Pull in chemo and bioinformatics, better data and information on food composition, the use of NAMs, expanded risk and classification trees. We know that the use of the threshold of toxicological concern has real value as part of a safety assessment, so we're looking to expand that knowledge base and start to use that as part of a decision-making tool, and then, of course, predictive toxicology. All of these things together can help reduce the use of animal testing, help predict potential hazards, and really improve timeliness so that we can get more of the substances that are out there evaluated and make sure that we are positioned to deal with the increasing volume that we see coming. So with that, I'll come to conclusion. Thank you for your time. We think that we should be using new data and information from NAMs, but it should be fit for purpose for regulatory use. We don't want to prematurely incorporate or rely on methods that have not been demonstrated to be fit for purpose. I think this is really important as we move forward at this pace where we're seeing more and more methods developed. New data and information should be considered along with existing data. We have a lot that we already know. We want to make sure that we don't forget about that and we incorporate that as well. We do consider process, but really as it affects the properties of the material itself. We don't regulate process, we regulate products. And fundamental for a strong pre-market program is early engagement. I didn't spend a lot of time talking about this, so it might be odd to have it here on a conclusion slide. But through the pre-market programs, all of them, we do have mechanisms that industry can come in and talk with us about what they're working to develop, what processes, what methods and protocols and approaches they're taking to safety. And this is really critically important because it helps make sure we have the information that we all need globally to ensure that substances that are coming onto the market have met that rigorous safety standard, the reasonable certainty of no harm. And with that, I'll thank you for your time. It looks like I still have a minute or so, so maybe if there's a question, thank you for having me and looking forward to the rest of the meeting and meeting many of you. Thank you.
Questions for Christy? Yeah.
Christy, thanks for that. Lee Neal here from Kazan's Food Standards Australia and New Zealand. To what degree in your work do you have regard to international standards or assessments that have been performed elsewhere?
We're certainly paying attention, I can tell you that. It's helpful wherever there is a validated or qualified method, especially as it relates to our food safety. So in terms of international assessments, we look at them, we review them, we pay attention to what other authorities are saying. Often we would agree, not always, right? In terms of standards that are out there, especially standards in terms of methods, I think it's really useful where we as an international body can come together. We pay attention to and participate in OECD specifically. We're also heavily involved in the WHO through the JECFA and the JMPR, where they are also setting methods and doing risk assessments. So I think international cooperation is critically important, and we are at the table and would like to stay there.
Hi, I'm Ana Afonso. I'm the head of unit here in EFSA for the Food and Nutrition Innovation. And my main question is, what do we do on the midterm? Because I totally agree with you, we need fit for purpose, we need to keep innovation going. And what I guess you are and I am is facing a situation where the rules we have, we cannot do it anymore, we cannot do our job. I'm thinking about things like intractable proteins. How do you follow the regulations that we have in place? I mean, what we do in the meanwhile, where we don't have those methods, but we can no longer use the ones we had before.
Well, I think there's certainly value in the ones that we have before. We don't want to throw away long-standing methods that have proven to be useful. So we certainly want to make sure that we're continuing to incorporate those. At FDA, we're a science-based organization. And so this is the exact question that you're raising, really presenting challenges. And I think that's why it's important that we identify what the gaps are and we move quickly to help validate or qualify. Actually, let me say qualify. Validate is such a big word, especially in this room, that we qualify methods to say that this is useful. Because right now, it's really taxing our experts. Right? Our experts are the ones left dealing with this, and they're doing it on a case-by-case, this study against this study. This is really putting us in a challenging situation.
I've got a question, Christy. So I think it was mentioned yesterday that, a lot of these alternative methods have been developed in the drug space. So now we're talking about food additives and food safety, and some of these food matrices are extremely complicated. So, what are the challenges there with trying to take data, that have been, I guess, developed out of these food matrices and trying to make sense out of it? Because as you said, we have an intended use. You're looking at a product for intended use. We as humans, we like to experiment. We may have a product that, you're supposed to consume it like it is right off the shelf. And we think, well, would this taste better if it was heated up? Maybe I'll microwave it. Maybe I'll add some water. Maybe I'll boil it. And so now a number of things could have happened, within these complicated matrices. So kind of address the challenges there that you're dealing with trying to take that data. What specific data are you looking for?
Yeah, I think that where you're talking about something that might had been evaluated or considered safe under one use condition and then expanded to another use condition. So I think there's a mix here of a little bit of a compliance issue, a human experimentation issue. I would say the human experimentation issue, if we can speak in generalities, which is difficult because humans are not general. But if we were to, we would say that the use conditions try to take those into account, right? So, should this plastic be microwaved? We assume that plastic is going to be microwaved, right? So we take those into account. It's not just what they say is the intended use. It's also what we would expect is practical based on that material. And so that gets incorporated into the original authorization. If industry is going to be changing their intended use conditions, it is their responsibility to come back in. And so that's where the compliance piece comes in. And that's, I think, why it's important that we all really understand the framework in the US that we're operating under, why we want to get out there and talk to industry so that they understand why we put out the guidance on what significant change is, because there could be expanded use from an industry perspective, and we would need to reevaluate that.
Follow up with the question outworks, yes. What do you see as the challenges that will be coming with the circular economy that we'll be facing over the coming decade and beyond?
Of the what economy?
The circular economy,
circular.
yeah. Sorry.
Well, I don't know. Maybe this is a little bit, I think, beyond the scope. But I think we understand. Maybe this is why we are looking and seeing more come into the recycling program as we're looking at these things. I think this is one of the future challenges that we're going to be dealing with, and this is why we're all talking together about, well, what are we doing here as it relates to recycling, reentry, and different sources of exposure, and these are all things that we are looking at and considering, and this is where we have more work to do.
Yeah. Thank you.
All right.
Okay.
Thank you, Christy.
Thank you, everyone. Thank you for your time.
So our next speaker this morning is Dr. Thomas Hartung. Thomas is the Dorenkamp-Zbinden Chair for Evidence-Based Toxicology in the Department of Environmental Health and Engineering at Johns Hopkins Bloomberg School of Public Health in Baltimore. He's also the director of the Centers for Alternatives to Animal Testing. His talk this morning will focus on tox AI-cology. AI is the future of toxicology. Thank you, Thomas.
Thank you very much. Good morning. This presentation was done by me, not by AI. You will hear these type of disclaimers more often in the near future, as you will see during my talk. I'm an academic, but I was working 7 years for the European Commission, actually here in Italy. And that's infectious. It's more difficult to get rid of than long COVID. You don't stop thinking about the regulatory use of methodologies. And so I will talk a bit about what from an academic perspective is happening at this moment. We are approaching the Christmas season, and my toxicologist Christmas wish list is pretty long. I wanted to really know something about human hazard, human risk, and so on, and I want to know it essentially for all of the chemicals which are around. The methods we are using in toxicology, in regulatory toxicology, most of them have been introduced when I was not yet born or in kindergarten. And it doesn't matter how young I feel, it's a pretty long time and very unusual, because these animal methods, sometimes they give us some ideas. We heard something about the brave new world of food safety, but there's about 10,000 substances which are relevant to food in the US. According to some studies, 80% don't have adequate data, and with the turnover of the system, it takes some 40 years just to get the backlog of these things done. And we have really no answers toxicologically for mixtures, for inter-individual differences in humans and so on. So I think we should at least agree that there is a lot of room for improvement. And the good thing is that this year's Christmas season is actually promising to be somewhat different, because there's a new kid on the block, which is AI. For many of you, AI might feel still a bit gimmicky. It is something nice to play around with, but I think most people don't grasp that over the last 10 years, AI has been doubling in capacity every 3 months. So our AI of today is about 8 times more potent than the AI of last year. And this is something we are witnessing. ChatGPT, everybody talks about this and knows about it. Last year, it did perform at the 10th percentile of a simulated lawyer's bar exam. So at the end of your studies, when you're really fit, 10% of the lawyers were worse than GPT. This year, it is at the 90th percentile. So it is really something which is enormous. And whatever you think about lawyers and their intellectual capacities, it is enormous. And this is all fueled by big data, because very simple, more than 90% of all data in the world were produced in the last 3 years. That's the number. These are numbers which are very difficult to grasp. And AI is making big sense of big data. That's the simplest definition which we have around. This enormous increase is possible because of the synergy of 3 technologies: data generation, the computing power. Moore's law said every second year our computers are twice as powerful at half the price, and this has held for 60 years. And AI, as I said, doubling every 3 months for the last 10, 12 years. So it is more than a billion-fold increase in power since we started to talk about AI. And this is really a disruptive technology which does not leave any area of our life unaffected. And reminds me of one of my favorite songs from R.E.M. It's the end of the world as we know it, and I feel fine. And I will show you why. There's a lot of miracles which have happened in recent years. Things we could not really believe they would happen. They seemed to be impossible. I will just talk about the first point, because this was for me an epiphany. When DeepMind in 2022 showed their model AlphaZero to play chess, it was really remarkable, because this model was never trained on a human game. They gave them 4 hours to train it and just to explore how can you play chess. And after 4 hours, it was as good as Magnus Carlsen, the current world champion. at I think 2,000 something, 2,600 ELO points. After 4 more hours, it was at 3,600 ELO points, unbeatable for a human being. And why I'm putting this up, not because I think you're interested in chess, but the interesting point was that it plays differently. It does not play like human players play. It sacrifices much more figures. It is looking for strategic advantages. And it is a situation now that human players study how AlphaZero is playing chess in order to improve their game. And this shows you that we should not expect AI to just come up with something on scale, taking over, doing the things like we have been doing them, emulating our intelligence. No, it is also coming up with new solutions. It is something which we have to take into account. AI comes in many flavors. People are thinking at the moment mostly of these large language models, which are really important because they are a prime data retrieval tool. We have been seeing that there was an interesting study by Christina Ruden about 20 years ago. She showed for 27 risk assessments for trichloroethylene that on average, these risk assessments were using 18% of the data which were available at the time, which were relevant for this risk assessment. This shows that we are typically not retrieving the data, and AI is perfect in doing this. Whatever is publicly available, this type of models can find. But it's not only about these multimodal data retrieval tools and then data integration tools. It also comes with many other flavors as expert systems, giving advice, helping to plan and optimize robotics, and most importantly for us as a predictive technology of machine learning, helping us where we lack data. And there's a lot to be said about all of these. These data extractions are really at the moment at the forefront. What I found very interesting is that for most of these models, we don't really know what they were trained on. These are data for BART, Google's model. 9% of all the data it was trained on is from health and medicine, science and health, 9%. What I found most interesting, the 2 most important sources for training these models were 2 open access online publishers, PLOS, Public Library of Science, and Frontiers. And this makes perfect sense, yeah? And this means in the future, if we want our science to contribute to the common knowledge, to be part of this, we have to publish open access. We have to stop these paywalls, because if we don't share with the world cannot digest it, cannot make use of these type of things. These improvements are enormous. This is the size of these figures for these large language models is showing the number of tokens, the variables they were trained on. GPT-4, when it came out on P-Day in this year, it was trained on about 100,000 times more variables than its predecessor, GPT-3. This is the explanation why it is performing about 10 times better. It was the same data set, actually. And this is a graph which shows how quickly these technologies are taking over human performance. It is getting faster and faster. And the last few graphs here in green, they are showing grad school math, code generation. Coders were a year or 2 ago one of the hottest commodity in the industry. Last fall, when the tech industry in the Bay Area did lay off 13% of people, they laid off 20% of coders. Because already last year, 40% of code was produced by large language models. This year, it is about 80%. What we need now is people controlling, quality assuring code, but not doing the coding work. This is something which is dramatically changing at this moment. What we have seen is that these models are, at the moment, for some very general uses. But from already 2017, when the bidirectional encoders were revolutionizing the world by Google, and you're using AI since then, because AI is in every of your Google searches, and you're trusting it, and it is not a question whether we use AI, it is only a question how we use AI, because nobody, I think, is not using any type of these browsers. Already then, we learned that we have to pre-train it for our users. We need to train it for the context of use of biomedicine. In February of this year, BioGPT was released by Windows, which is helping AI to read scientific literature. What I found most interesting, BioGPT is outperforming humans in annotating scientific papers. What does it mean? AI is reading scientific papers better than your PhD student. But it's not reading one paper a week, it is reading millions, whatever you can feed into it, and it's not going to forget. So this is an enormous potential we really have to see. We are probably the last generation which was trained to write a scientific paper from scratch. This is from a venture capital company, and they assess that in 2023, now, we're already getting good scientific drafts by these type of AIs, and they calculate that in 2030 latest, we will get something which is better than a professional scientific writer from AI if we draft our texts. This is something we simply have to face. It's nothing we can avoid, and this was an interesting view in Nature. Open generative AI models are a way forward for science, and I'm with this comment because it is like when handheld calculators were introduced in my time in school, we were discussing whether kids would stop learning mathematics. And today we would smile about this, but it is simply the way we are using our tools. This is an article by my friend Jean-Claude Burgelmann. We worked together in the commission, and I was asked to comment on this article about regulating AI. And I did an experiment. In my response, I simply let ChatGPT write my answer. I said, summarize, appraise, and criticize the article. And I put this article, the answer, as my supplement to my own article. And this was a very interesting experience because Jean-Claude and myself, we both agreed both articles were good responses. I could have, without problem, just submitted what GPT-4 was giving me. It was, however, something which was rock solid, but it was a bit lame. It was just giving the most probable answers, and it forced me to focus in my own article on ideas, some quotes, some out-of-the-box thinking. And I think this is something which will happen. Yeah? We humans have to think about what can we do above just the lame summary of what everybody could know if they only read the scientific literature. So I believe we are entering a phase of toxicology, as I put in the title, because we are, it is going to change us. And this is because we have big data, a discipline which has been very poor in data, where we counted dead animals or the number of tumors in organs, something you could do perhaps even without a handheld calculator, is moving to high content, to high throughput, sensor technologies, an enormous information. There's about 20,000 articles in PubMed with the meshword of toxicology every year. About 800,000 of the articles in PubMed describe some effects of chemicals, not necessarily as drugs or toxicants, but as inhibitors, as actors in biological systems. We found 900,000 safety data sheets in the internet. Yeah? It's not the best scientific information, but there is a lot around this. And the big computers, the AI and machine learning, is at the moment developing so fast. It is not only about the power of them, but also the fact that we are now using foundational models. You don't have to write your new AI model. You just integrate your problem and your data in existing models, and you get, in addition to the most powerful infrastructure, you get all of the information which is already in, helping you, often as weak associations. And this makes big sense. And this big sense is, first of all, about data retrieval. We really have access to the things which are findable. It is helping us increasingly to integrate this evidence, whether it is in systematic reviews, in our risk assessments. It is predicting and filling the data gaps which we use. We have seen in pathology, which was a little bit faster than toxicology once again. And what we see is how the interpretation of pictures, the storage, the comparison, the interpretation of these is a revolution as digital pathology at the moment taking place. And it's also increasingly helping us to write all of these dull reports because it is giving us in minutes something we have to fine-tune and quality control, but is something which is making our work faster. We have in 2018 published this work, which got a lot of attention at the time because it was an early use of AI in this field. We used at the time 10 million structures, 900,000 of these had some data, very different labels which we were using. And what we observed is that we were able to predict 9 OECD guideline type of tests with 87% accuracy. And we showed this for 190,000 chemicals. Yeah? Imagine this, 190,000 chemicals. This was just by number crunching. And this compared favorably to an overall 81% reproducibility of these animal tests. So it was better than the animal tests we are using to come to these classifications because it does what a good toxicologist is doing. You not just take the result of the animal test, you use all of the information you have and you integrate it and come to a conclusion whether you can trust the result or not. Because we were using 74 different labels. So what's happening here was enormous network effects because these things get powerful by big data, and it was transfer learning because if you are interested, let's say, in eye irritation, it is stupid just to look for information on eye irritation. You might know nothing about the substance and neighboring substances, similar substances, but you might know something about skin corrosion, skin irritation. And something which is a skin corrosive is obviously also an eye irritant, or something which is a genotoxicant is probably reactive chemistry. So this is also informative for these other endpoints, and that's boosting accuracies. So from this, we have moved on. In 2020, we showed for skin sensitization that it's also predicting human data pretty well. It was for 506 chemicals, 80% correct, while the local lymph node assay, as the favored animal model, just gets about 74% of human skin sensitizers. Interesting for you, I hope. We predicted 4,700 food-relevant chemicals in the US in this study, just to show that this can be done on scale. In less than 1 hour, we predicted for each and every of these chemicals the 9 outcomes which we had in our model, which is the equivalent of 38,000 animal studies, which would cost you more than $250 million to execute if you only find the laboratories to do this all. And in 1 hour, we got something which was, I have to admit, on a very small scale, where we carried out a validation here, 83% of the time correct. Again, pretty good, at least as good as the animal models would be. So we could have, if we only wanted it, the entire chemical universe evaluated probably within days, probably even hours nowadays. Yeah. And we showed at SOT this year that this is not limited to the acute and topical toxicities. We took data from the REACH registration for reproductive toxicity and for cancer. And again, we had pretty good predictivities of 75 and 82% of the classifications which were done on this basis in the REACH process. This is far better than the reproducibility of these systemic toxicity tests, which is only in the 60% range if you repeat some of these animal experiments. There's a dark side to AI. I don't want just to say the world is beautiful, brave new world. It has enormous data needs, and biases in the data sets will always be a problem. We have difficulties at the moment to explain the results, causality, validation. These are certainly big challenges. However, we're making a lot of progress. First of all, AI is not an autonomous system. I don't suggest that we use AI as something to replace any toxicologist. The human has to be in the loop. We have to quality control these things. We have to train these models. But we get information and evidence integration now on a silver platter. And we're making enormous progress at the moment on explainable AI, where the AI is saying why it came to some conclusions. It is no longer the black box. It can really identify and explain itself. At the moment, this is associated with some loss in accuracy, but I think this is only a matter of time to do so. So really have to see that this is an enormously developing area. We are at the moment in the age of I learn to learn, where we let the system try and gets better and better, improves the reinforcement learning. But we're already seeing at the moment how these systems are starting to talk to each other. They are federated, they are working as swarm intelligence and are approaching more and more areas of the world. And this is very relevant also for us, where the majority of data is behind firewalls. Most of the data in toxicology are owned by companies. And you will have a long fight with lawyers before they will be released to train our models. But we can send our algorithms, they can run on this data behind the firewall, and then we get a better trained algorithm back. No data. So this is really opportunities of sharing information and making our predictions better. Another trend at the moment is these foundational models. I already mentioned this. You don't need a coder to write your new AI. You can really integrate your problem into the existing models with the same beautiful user interfaces, and you can start using them and benefit from all of the information they have already been trained on. We all know that in the US, there was a watershed moment in 2007 when this report, Toxicity Testing in the 21st Century, was published. And I've been working on the implementation of this report since. This is about 14, 15 years ago, and science is doubling every 7 years at least. Some people say it's by 2 to 3 years at the moment. So we have now about 4 times more knowledge than at the time of this report. So I think it is time for a version 2.0 of this. And you have to simply see induced pluripotent stem cells, which are giving rise to microphysiological systems, were invented in 2006. They are not part of the report. Omics is not part of the report. AI is not part of the report because these technologies were not around. I had the privilege for the Department of Defense to lead a workshop exercise to think about a toxicology 2.0 type of report. And this is the report which we published in June of this year, so it's available. It's an overall a call for a human exposome project in the sense of complementing the human genome with the exposure part of the exposure side of health and disease. And some 30 participants, 15 agency observers, they came up with this call. This is an article of mine which is elaborating a little bit more on the idea of the exposome project. It is saying really we have to be more exposure driven. We have to be technology enabled, embracing AI, omics, and microphysiological system evidence integrated. We are exploring a lot of this at the moment in a large-scale project by the EU ONTOX project. This project, coordinated by Mathieu Winken, brings together 17 groups which are over 5 years exploring, among others, what AI can deliver, what we can do for liver, kidney, and the developing brain toxicology. It is based on data inputs. One of the things is, again, work by Tom Lichtefeld. He developed sysref.com, which is a tool which is freely available to carry out systematic reviews faster. This tool has been used for more than 10,000 projects in assessing literature, and it's based on machine learning. It is learning from you which papers to include, and after a few hundred papers, it is as good as a second human in identifying papers. A second tool, so this is really auto-annotating. In the context of ONTOX, we are helping to extract the information now, also natural entity recognition, causal relationships. So we're feeding this into the process, taking advantage of BioGPT and others. The second tool we are using is databases. There is tons of public databases in the field. We call these BioBrick as an approach, which is more or less putting an automated one-line import function to each of these databases. And the third one is ChemChart, which is a meta-crawler of chemical information from the internet, which is able to read whatever is available for chemical information. This is the type of data. Let me say a word about these technologies. ChemChart It was not a very successful product, but it's extremely helpful for us now. It was designed by Tom and his team in order to get information for any chemical you would put in, which would tell you where can you buy it, who is able to synthesize it in the world, what are the known safety informations, what are alternative chemistries, what are the testing data which are available, and how it is regulated. And all of this goes in. And as I said, 900,000 safety data sheets is one of the things it identified. But let's talk about BioBricks for a second. This is a survey which showed that 45% of the time of a data scientist is for loading and cleaning data. 45% of the time of these heavily paid people. BioBricks are converting this process to a one-line command. You just say upload, and you get within a minute or two, you get the entire PubChem, Campbell, whatever you want, the entire registration data of ECHA, whatever you want to include. And with support from the NIHS, the National Science Foundation, and ONTOX, Tom and team have now produced 50 such BioBricks. So all of the publicly available databases relevant in our field can be included. ChemHarmony is one example. He used 8 of these, and he found in total 200 million data points where you have a chemical, a property, and the result. That's really something to train a system on. He used only 1 million of these data points because we are seeking at the moment the supercomputer capacities to really do all of these comparisons. And he was able to predict 155 properties of chemicals with more than 80% accuracy. And you can turn it around. You can say, these are the properties. Tell me which chemistry does fulfill it, whether it's already synthesized or not. It is really enormous what these models can do. And I think there's a lot to be expected. And part of this is now also to bring in, beside the chemical similarity, the biological similarity. So you see here, you can be structurally similar or you can be biologically similar. And this is what we are trying to bring into the system. This has then enormous consequences. By structural similarity, he achieved, for example, about 60 to 70% accuracy over these 152 properties. If he was using biological one, as I said, it was more than 80%. And the computational power is getting better and better. But it cost us about $5,000 in 2018 on an Amazon cloud server with 180 cores. It's now done by a reasonably normal computer. Our computers are about $20,000 a piece, and they do 1 trillion comparisons per hour. The problem is if you have 200 million data points, this is about 20,000 hours for all of the pairwise comparisons which you have to run. It shows you the challenge also computationally to run something, but it is not really a problem for a supercomputer. We have to do something with the data. The ONTOX project is feeding them in a semi-automated way into physiological maps and adverse outcome pathways. So we illustrate what we find in the internet for these points. So in the end, we are trying to develop at the moment a concept of perturbation of physiology to lead to us to a probability of hazard as chemical structure and the biological properties can do. And probability is really the magic word here. Because the big problem I see we have in toxicology is that we are black and white. We like to have our classifications of this is toxic, this is not, this is carcinogenic, this is not, while the reality is delivering a lot of shades of gray with some uncertainties. And as soon as we start adopting this view, this has tremendous impacts. So we have reassessed the concept of probabilistic risk assessment under the viewpoint of AI. Because from my point of view, probabilistic approaches only become beautiful by AI. The idea behind this is that AI is delivering probabilities. It is never certain. A lot of this is wrong, but it is delivering the most probable result based on the data it has been trained on. So in the ONTOX project, we are trying to develop a model which is doing a probabilistic risk assessment, which then shall allow us also to do information economy. You can ask, do I really have to test more? Would it change the assessment? Would it really change the entire landscape? Is there really one test which can do all of this, given all the information we have? And if so, what would be the test which is really changing our views? And then we can recommend targeted testing. We can say, okay, this is a way of using a microphysiological system or something to do this. I would like to end with one of the most exciting developments at the moment in my scientific life, which is combining AI with biological systems. This is a brain organoid. We have been working on these models for the last 20 years. We were the first to mass produce brain organoids in 2016. And we're combining now brain organoids with AI through electrophysiology. And the output of this is what we call organoid intelligence. We believe we are showing at the moment learning and memory in these systems. Our partners from Cortical Lab have already shown that neuronal cultures can play the computer game Pong. We are jointly using now brain organoids, and I think we are able to show at the moment long-term memory, rewiring of these structures in response to inputs. And this has enormous impact on physiology of learning, brain-machine interfaces, finding things in diseases like Alzheimer. Alzheimer is increasing more than the aging of the population would suggest. What are the lifestyle factors which are impacting on this? Yeah? We have no systems really to study this, but we see that brain organoids from Alzheimer patients are showing some of the hallmarks of Alzheimer. So we published this concept in February. There were more than 600 press hits instantly. The US White House has taken this up as one of the bold biotechnology goals for the US to produce organoid intelligence. National Science Foundation has taken up this idea and is now working out an engineering organoid intelligence program, and we are developing something which we call DORI, the Dementia OI Research Initiative, because we are trying to use this for Alzheimer and similar. So let me come to a close, and my slides are available. By the way, this picture was produced with DALL-E, with AI. That's the only component in this talk. I think we are at a time where we, for the first time, can really integrate scientific knowledge with AI. AI is the answer to information flooding. It is really condensing information and putting it back to a manageable amount. Nobody can read 20,000 papers in toxicology per year. It is accelerating drug development. Last year already, 18 AI-first chemicals went into human trials, phase 1. This year, the first of them went into a phase 2 study. So it is something which is coming. It is optimizing public health, prevention, something I'm very much interested in as a toxicologist. I think most of you are. And it's democratizing healthcare access, because you have all of the knowledge of the world at your fingertips. You can use your phone and ask it what is known about this disease, and if you don't have access to these type of people. We have a path forward which is open access, fair data, machine-readable stuff. There's a challenge in identifying bias in the data, but if human can identify bias, machine can too. We only have to train the machines. And there's a deficit at the moment in explainable AI, but this is coming. We are having better and better methods to have the AI explain why a certain probability was achieved, what were the most influential things taken into consideration after being trained on millions and billions of data points. Thank you very much.
Thank you very much, Thomas, for a very inspiring presentation. We have time for a couple of questions. Wida?
Hey, Thomas, always enjoy your presentation. I just want to pick up the last line you mentioned about the explainable AI. Well, when we talk about the explainability, basically means something can be explained, understood by humans. But what you, at the very beginning, you mentioned the AI really wants to succeed at the humans and make a certain move which humans not necessarily understand. So there, I just want to get some source from you. How important the explainability in the AI?
I think you have a very important point. The problem is that what do we mean by explainable? We want to see our biases reinforced. We want to see that the things we believe are actually shown by the system. whether this is right or not, I don't know. Because it is a bit inbred. We don't allow the AI to come up with new solutions if we say, show us that you make the same mistakes we have been doing over the last decades. But it is at the same time helpful if you can ask it what was informing your choice and the things you're doing. In some instances it's easy. If you used early forms like random forest, and so you can see what were the decisions taken, what were the thresholds, what was informing a system. And this is very helpful. In our 2018 paper, we showed exactly what is intuitively clear, that eye irritation was informed by skin irritation or by mutagenicity, which makes sense, and this gives face validity. But I agree, probably we would not be better than Magnus Carlsen in playing chess if we expected the system to show that it's playing like a human. And this is something where we have to perhaps open our minds, but at the moment in a transition phase, I think it is extremely helpful if we get mechanistic thinking into this, if we show that some of the components make sense. This builds trust.
Yeah, thank you very much. Very inspiring talk. My question is quite simple in a sense, like I fully agree that an AI certainly can also recognize bias. So if a human can do it, why not the AI? But what if the majority of the publicly available data is just biased? Then how to approach that?
I mean, I have a problem. I'm arguing against animal testing, and I say we use all of these data and make wonderful results from it. Yeah, that's a problem. We have somewhere to start, and it also will require data generation. I think that, for example, the ToxCast project or the Tox21 project in the US, which are enormous data generation exercises, are most valuable, not because they are predicting anything in toxicology, but they are characterizing chemicals in hundreds of assays and are delivering this for thousands of chemicals in a very standardized way. And these type of data generations we will need. But AI also can help us to tell us what are the nodes, what are the understudied structures, what are the things we should actually address and produce data on. And I think this is something moving forward. It is always good that these tools are not only telling you what we can know, but also what we cannot know, where the probability of a judgment is so poor that you simply don't want to trust it. And these are the only methods which tell you, I came to this judgment and it is 70% probable. And then you can decide this might be good enough for a household cleaner or an intermediate chemical in a production process, but you would not like to do it and put it in baby food. So it is really allowing you to say, oh, we need to study more, or something which is absolutely certain, because it is not comparing to a single chemical for your read across, but it's comparing to the chemical universe around it. Then you come to something where it is so unlikely that the $1 million cancer study is going to give me a different result that I simply don't do the study and simply classify the substance and say it's precautionary to just use the classification.
Thank you very much. We have time for one final question.
Okay, thank you. Well, thank you very much, Thomas, again. Lovely to hear you again for the second time on this. I have one comment on when we're talking about ChatGPT and all of these things, are most of the time they're semantic. So we understand the logic, language logic behind it. We know that we are in the logic of the first order of logic is very tight. The upper and lower, I would say, limits are very well understood. Mm-hmm. What I want to know from your side in future, because it's very interesting what you're doing, how can we tackle the problem with the lower and upper limits of the physiological and inter-individual differences that we would have? Do you have any thought about this? That would be very interesting to see where you're going.
Yeah. I mean, we need the data before we can expect the answers. And if we don't have enough data about inter-individual differences, no system in the world can give us reasonable answers. So we really need to think about data generation processes and how to link a lot of the information. But at the same time, a human genome, the first one was 3 billion. Now we get a full human genome for $200. There's now enormous opportunities in interpreting these things, and but the genome is not everything. We need to combine this with the consequences, the phenotype. And there's more and more studies, all of us in the US and many biobanking studies in Europe. There's more and more information. We have to make it machine readable and interpretable. We should also all be aware how early we are in the deployment of these technologies. Last year's GPT-3 was at the 10th percentile for mathematics tests. Yeah? This year's GPT-4 is at the 90th percentile. Yeah? It's better than most high school students at this stage. And we have to expect that GPT-5, which is probably released around the turn of the year, is 100 times stronger than GPT-4. And I'm not advertising OpenAI and these things. It's just showing the enormous development, what we should expect, and we should stay open for this. We are missing a chance if we are not adapting these, but we're also doing a mistake to think that this will be autonomous and taking our jobs. No, we have better work and better throughput with using these technologies as a co-pilot.
Thank you very much. So this brings us to the end of the first part of session 1, and we're breaking up for coffee break. We should be back here. We'd like to be back here at 11:10, please. 11:10.